Hi my dear student, welcome to my channel. Today discussing about the topic of asexual and sexual reproduction in plants, part 3. In the previous class discussing about the topic of asexual reproduction in plants. Today discussing the topic of sexual reproduction in plants. In this class, only discussing about the male reproductive part, that is andrecium. The sexual reproduction means involves in the production and fusion of male and female gametes. The former is called gametophyte. Later is the process of fertilization. In algae and bryophytes, they reproduce by the production of gametes, which may be motile or non-motile depending upon the species. The gamete fusion is of three types: isogamy, anisogamy, and oogamy. Isogamy means morphologically and physiologically similar. Anisogamy, morphologically similar but physiologically dissimilar. Oogamy, both morphologically and physiologically dissimilar. In algae, external fertilization takes place, but in higher plants, internal fertilization occurs. The next discussing the topic of the reproductive organ. The flower is the only the reproductive organs. Flower consists of 4 parts: calyx, corolla, andrecium and gynoecium. Flower is multi-dimensional perspective from the time immemorial. It is an inspirational tool for the species. It is a decorative material for all the celebrations. In Tamil literature, the 5 lands are denoted by different flowers. The flag of some countries are embedded with the flowers used in preparation of perfumes. So a flower possesses 4 holes: calyx, corolla, andrecium and gynoecium. Andrecium and gynoecium are essential organs. The process or changes involved in the sexual reproduction of higher plants include 3 stages. Number 1, free fertilization. Number 2, fertilization. Number 3, forced fertilization. So the first changes involved in the reproduction. Number 1, free fertilization, structure and events. The hormonal and structural changes in plant lead to the differentiation and development of floral primordium. So the male reproductive part, that is called andrecium. You know, andrecium and gynoecium are essential organ. So the andrecium is a male reproductive part. The andrecium is made up of stamen or microsporophyle, male reproductive part of the angiosperm flower. Each stamen consists of 2 parts, namely a filament and an anther. Monothecous, when the anther consists of a single lobe. Example, the members of the family Malvaceae. Dithecous, when the anther consists of 2 lobes. Example, members of the family Rutaceae. The stamen, the 2 lobes of the anther are connected by a tissue known as connective. Each lobe of the anther contains 2 pollen sacs or microsporangia. Thus, there are 4 pollen sacs in each anther. Each pollen sac contains numerous fine powdery mass of cell called the pollen grains. So the next one, development of anther. The anther is the part of stamen producing microspore and pollen grains. The stamen or microsporophyll arises as a small outgrowth of the thalamus. It consists of meristematic cell, it becomes differentiated into lower slender portion called filament and an upper portion called the anther. The young anther consists of a homogeneous mass of meristematic cells. It is surrounded by a epidermis. The epidermis is single layered. In dithecous anther, the anther soon becomes four-lobed, and in each lobe a microsporangium develop. In each lobe of the anther, a plate of vertical row cell get differentiated to form archisporium. The archisporial cell are hypodermal in origin. They are large, radially elongated, and contain dense cytoplasm and prominent nucleus. The archisporium is formed of a single cell, as in Boyer-Havia. The archisporial cell divide periclinally to form two layers of cells. The outer layer is the primary parietal layer, and the inner layer is called the primary sporogenous tissue. The primary sporogenous tissue develop into pollen mother cell. The pollen mother cell undergo meiosis to produce haploid pollen grains. The cells of the primary parietal layer undergo a series of periclinal and anticlinal division to form the wall layers of the anther. So next discussing the topic about transverse section of mature anther. In the mature anther, the wall is formed of epidermis, endothecium, middle layer, and tapetum. A typical anther is a tetrasporangiate. It contains three major parts: anther wall, anther cavity, and connective. So the number one, anther wall. Anther wall contains four layers are there: epidermis, endothecium, middle layer, and tapetum. First one, epidermis. Epidermis is the outermost layer of the anther. It consists of a single layer of cell, and the cells are compactly arranged. It exists from the very early stage, and it is not derived from the primary parietal layer. The cells of the epidermis undergo anticlinal division, and so the epidermis increases the surface area. In matured anther, the cell of the epidermis are greatly stretched and flattened. Epidermis is an protective in function. So the second layer, endothecium. Endothecium is the second layer of the anther wall, is also known as fibrous layer due to the presence of fibrous band. It is present just below the epidermis. It is formed from the primary parietal cell. It consists of a single layer of radially elongated cell. The endothelial cell show the characteristic fibrous band of lignin which arise from the inner tangential walls. The cells of the endothecium along the line of dehiscence of the anther and divide of the fibrous band. These cells are called stomium, where the endothecium is thin-walled. The stomium helps in the dehiscence of the anther. The endothecial layer become hygroscopic due to the development of fibrous band, and they play an important role in anthesis. Endothecium absent in anther whose dehiscence is through apical pore, as in cassia and solanum. Fibrous band are absent in annona, musa, and hypomea, etc. Third one, middle layer. It is a third layer of the anther wall lying below the endothecium. It consists of 2 to 3 layers of thin-walled parenchyma cells. It is also known as parietal layer. The cells of middle layer store starch grains. The middle layer cells get crushed at the time of meiosis in the pollen mother cell. Fourth layer, tapetum. Tapetum is the innermost layer of anther wall. It surrounds the sporogenous tissue. It is formed beneath the middle layer. The cells of tapetum are bigger, radially elongated, with the dense cytoplasm and prominent nucleus. It is a nutrient layer for the developing pollen grains. Usually the tapetum is parietal in origin, but in Electrothompsoni, the tapetum is dual in origin. A portion of the tapetum is derived from cells of the connective tissue and are called Xii tapetum, and the remaining portion of the tapetum is derived from the parietal cell and are called P tapetum. Thus, there are two types of tapetum, namely Xii tapetum and B tapetum. The cell of the Xii tapetum are larger and the cell of the P tapetum are smaller. Thus, the tapetum with two different types of cell is called dimorphic tapetum. So, the types of tapetum based on the behavior of tapetum is two types: glandular or secretory tapetum, amoeboid or plasmodial tapetum. Number 1, glandular tapetum. It is also known as secretory or parietal tapetum. In a glandular tapetum, the tapetal cell remain intact in their original position throughout the development of microspore. The tapetal cell do not degenerate. The cell become rich in protoplasmic content and secrete a plasmodium that surround the pollen grains. Secretory or glandular tapetum is common than the amoeboidal tapetum. It is found in the anthers of Helleborus poetidus, Mirafilis jalafa, etc. In Helleborus poetidus, Eglin and Godwin have recorded the formation of ubish bodies in the tapetal cell. These bodies contribute to the material known as sporopollen, which is involved in the thickening of the exina pollen grains. Second one, amoeboid tapetum. It is also known as a invasive or periplasmodial tapetum. In amoeboid tapetum, the inner and radial wall of the tapetal cell break down and the protoplast become a plasmodial mass called tapetal periplasmodium, in which the pollen mother cells are suspended. Amoeboid tapetum is found in the anthers of Alisma, Typha, Tradescandia, and Butamus. In both amoeboid and glandular tapetal cell, total DNA content increases during meiosis. This phenomenon occurs in the following method. Number 1, multinucleated condition. Number 2, endomitosis. Number 3, sticky type of mitosis. Number 4, polyteny. As a result, the tapetal cell may be uninucleate or binucleate or quadrinucleate. These are about the important anther wall. Number 1, epidermis. Number 2, endothecium. Number 3, middle layer. Number 4, tapetum. So the one question will be asked, list out the important function of the tapetum. And a tapetum is play a vital role for many purpose. Tapetum is a nutrient tissue. In the developing microsporangium, the tapetum forms a channel for the transport of material to reach the pollen mother cell. The tapetum serves as a nutrition of the pollen grain or microspores. Tapetum play an important role in the pollen wall formation. The ubish body present in the tapetal cell contribute a material called sporopollen, which is involved in the formation of exina pollen grains. This has been demonstrated in Helleborus poetidus by Eglin and Godwin. The plastids of the tapetal cell synthesize materials like pollen kit and trypsin, discovered by Dixon 1978. This material protects the pollen from damaging effects of ultraviolet rays, and its sticky nature helpful in insect dispersal of pollen. The enzyme callase, which derived from the dictyosome of the tapetal cell, degrade the callose around the pollen tetrad, and hence the pollen grains in the tetrad are separated. This has been demonstrated in Tradescandia by Mepham and Lanny in the year 1969. Tapetum is essential for the formation of viable pollen grains. Tapetal degeneration during meiotic stages causes male sterility in petunia. The proteins present in the axon of the pollen wall are essential for regeneration of compatible pistils. These proteins are derived from the tapetal cell. Tapetal cell store nutrients. So the typical mature anther consists of three parts: anther wall, anther cavity, and connective. Number 1, anther wall over. Number 2, anther cavity. It is filled with microspore in young stages or pollen grains are at maturity. The meiotic division of the microspore mother cell give rise to microspore which are haploid in nature. Third one, connective. It is a column of sterile tissue surrounded by a anther lobe. It possesses vascular tissues. It also contributes to the inner tapetum. Another one, microsporogenesis. The development of pollen grains or microspores from the diploid pollen mother cell by meiosis in the microsporangia of the anther is known as microsporogenesis. The pollen grain develops from sporogenous tissue of the archisporium. The sporogenous cell develop into pollen mother cell. The pollen mother cells are diploid and they undergo meiosis to produce pollen tetrad. The pollen tetrad get separated to form pollen grains or microspores. The pollen grains are haploid. The meiotic division of the pollen mother cell may be 2 types are there: successive type and simultaneous type. In successive division, meiotic divisions are followed by immediate wall formation. Example, Xii. In each pollen mother cell undergo burst meiotic division resulting in the formation of 2 nuclei. The nuclear division is followed by immediate wall formation. Thus, a dyad is formed. The 2 cells of the dyad undergo second meiotic division, which is also followed by wall formation. As a result, a tetrad is formed. In this type, cell plate is formed from the center and then extended centrifugally on both sides. This type of wall formation is common in monocots. Second one, simultaneous type. In simultaneous division of wall are formed after the formation of 4 nuclei. Example, Millilotus alpha. The wall formation in simultaneous type is centripetal. This type of wall formation is common in dicots. The orientation of the 4 microspore in the tetrad varies. Tetrahedral is an example very common. Isobilateral siamase, decussate magnolia, T-shaped aristolochia, linear calophylla. In Pollinium, Asclepidaceae and Orchidaceae family members, all the pollen grain in a sporangium remain together to form a single mass called pollinium. They may be corpusculum and caudicle. The main stalk is called corpusculum. The stalk is called caudicle or retinaculum. The bag-like structure is called pollina. So it is called pollinium. In 4 microspore of the tetrad fuse together forming a compound pollen grain. Example, Begonia, Drosera, Drymis, etc. Another one, pollen grains. The study of pollen grain is called palynology. The pollen grain is the bust cell of the male gametophyte. The pollen grains are formed inside the pollen sac of anther. It develops from the pollen mother cell by meiosis. They are haploid, unicellular and microscopic. The largest pollen grains are found in cucurbita. The smallest pollen grains are found in myosotis. It consists of a wall, cytoplasm and a nucleus. So the wall of the pollen grain, outer layer exine, inner layer intine. Exine is thick and is made up of cellulose, sporopollen and pollen kit. When these thin areas are small and rounded, it is called germ pores, or when elongated, it is called furrows. It is associated with the germination of pollen grain. The sporopollen is generally absent in germ pores. It is made up of two layers, outer extexine, that is called sculptured, inner endexine or endoexine. Indine thin, uniform and is made up of pectin, hemicellulose, cellulose and callose together with the proteins. Next one, the shape of the pollen grain. Shape of the pollen grains may be different shapes of there. Globose, ellipsoid, fusiform, lobed, angular or crescent shapes are there. Then size of the pollen grain, 10 micrometer from the myosotis plant. You see the picture. The size of the pollen grain is very small, in 10 micrometers in myosotis plant. Second picture, 200 micrometers. The first picture, Cucurbitaceae family. Second picture, Nyctogenaceae family. It's a very small in size. So 200 micrometers in Cucurbitaceae and Nyctogenaceae family. Next one, sporopollen. The wall material of sporopollen is contributed by both pollen cytoplasm and tapetum. It is derived from carotenoids. It is resistant to physical and biological decomposition. It helps to withstand high temperature and is resistant to strong acid, alkali and enzyme action. Hence it preserves the pollen for long period in fossil deposit, and it also protects the pollen during its journey from anther to stigma. Another one, pollen kit. Pollen kit is contributed by the tapetum and colored yellow or orange and is cheaply made of carotenoids or flavonoids. It is an oily layer forming a thick viscous coating over pollen surface. It attracts insect and protects damage from UV radiation. Another one, pollen bank. The pollen grains are stored in the laboratory condition. So palynology is the study of pollen grains. It helps to identify the distribution of whole and to locate oil field. Pollen grains reflect the vegetation of an area. Liquid nitrogen minus 196 degrees Celsius is used to preserve pollen in viable condition for prolonged duration. This technique is called cryopreservation and is used to store pollen grains, that is called pollen bank, or economically important crops for breeding programs. Another one, pea pollen. Pea pollen is a natural substance and contains high protein, carbohydrate, trace amount of minerals and vitamins. So the study of honey pollen is called melitopalynology. Another one, pollen calendar. Pollen calendar shows the production of pollen by plants during different seasons. This benefits the allergic person. Pollen grain cause allergic reaction like asthma, bronchitis, hay fever, allergic rhinitis, etc. Another one, pollen allergy. Pollen allergy is caused to the plant Parthenium chistiroporus. Family Asteraceae is commonly called carrot grass, in a native of tropical America, and was introduced into India as a contaminant along with cereal wheat. The pollen of this plant cause allergy. Another one, pollen tablet. The pea pollen is used as a dietary supplement and is sold as a pollen tablet. Pollen tablets are used by several purposes. Another one, pollen syrup. Pollen syrup is a dietary supplement of food also there. Further, it increases the performance of athletics, race courses, and also heals the wound caused by the burns. So the various purpose of the pollen grains. The pollen grains are several products are there. Another one, development of male gametophyte. The pollen tube containing the male gamete is called male gametophyte. The pollen grain is a burst cell of the male gametophyte. After it release from the anther, it undergo expansion, which is followed by vacuolation. As a result, nucleus is pushed to the periphery. The pollen grain undergo burst mitotic division to form two unequal cells. The larger cell is known as vegetative cell. Smaller cell is known as generative cell. The vegetative cell is bigger than generative cell. It has starch and fat. The vacuole of the vegetative cell disappear. The cell organelle like plastid, mitochondria, ribosome, and the RNA content and protein increases. The nucleolus is larger than the nucleolus of generative cell. DNA content is less. The generative cell at the early stages is hemispherical and remain attached to the pollen wall. Later, the generative cell detaches from the wall and becomes spherical. The generative cell is spherical and is smaller than the vegetative cell. The cytoplasm of the generative cell is highly reduced and highly in appearance. It lacks reserved food material and RNA. The DNA content is very large. It contains the cell organelle like mitochondria, ribosome, dictyosome, microtubules, etc. Another one, the pollen tube. The pollen grain shed from the anther usually in two-celled stage, that is vegetative and generative cell. For the development of the male gametophyte occurs after the pollen grain reaches the stigma. The indine of the pollen grain protrudes through one of the germ pore. It forms a delicate tubular structure called pollen tube. It was first discovered by Xii in the year 1823 in Port Lacca, Poland. The pollen tube grows through the style. The pollen grains may be monosiphonous or polysiphonous. When the pollen grain produces a single pollen tube, as in majority of the angiosperm, it is called monosiphonous. If it is produced more than one tube, as in malva plant, the pollen grain is called polysiphonous. The pollen tube is branched in cucurbita and papaya. The vegetative and generative cell of the pollen grain migrate into the pollen tube. As the pollen tube grows through the style, callous plugs are formed in the pollen tube at regular distance behind the growing tip of the pollen tube. The vegetative nucleus moves burst inside the pollen tube. Now it is called tube nucleus. The tube nucleus is assumed to play an important role in directing the growth of the pollen tube. The generative cell divide mitotically to form two oval or elliptical male gametes or sperm. The formation of two male gametes or sperm from a generative cell is known as spermatogenesis. In majority of the angiosperm, the generative cell divide to form two sperm just prior to the entry of the pollen tube into the embryo sac. This portion can be evaluated. Number 1, pollen tube was discovered by option A. J.G. Kohlreuter, B. G.B. Amisi, C. E. Strasburger, D. E. Canning. So the answer B. G.B. Amisi. Second question, size of the pollen grain in myosotis. A. 10 micrometer, B. 20 micrometer, C. 200 micrometer, D. 2000 micrometer. So the answer A option 10 micrometer. Third question, burst cell of male gametophyte in angiosperm is. A. Microspore, B. Megaspore, C. Nucleus, D. Primary endosperm nucleus. So the answer A. Microspore. Question number 4. Match the following. First one external fertilization, second one andrecium, third one male gametophyte, fourth one primary parietal layer. So the correct match: external fertilization, algae, andrecium, stamens, male gametophyte, pollen grain, primary parietal layer, anther wall. Fifth question: arrange the layer of anther wall from locus to periphery. Arranging the layer locus to periphery means center to periphery. Answer: tapetum, middle layers, endothecium and epidermis. Sixth question: differentiate between secretory and invasive tapetum. Seventh question: name the cell which divide to form male nuclei. Answer: generative cell. Eighth one: endothecium is associated with dehiscence of anther. Justify the statement. It is generally a single layer of radially elongated cell found below the epidermis. The cell along junction of the two sporangia of an anther lobe lack this thickening. This region is known as stomium. This region, along with the hygroscopic of nature of endothecium, help in the dehiscence of anther at maturity. Ninth question: list out the functions of tapetum. 10th question: write a short note on pollen kit. 11th question: discuss the steps involved in microsporogenesis. In this chapter, they are discussing about some technical terms. Endothecium, a single layer of hygroscopic, radially elongated cell found below the epidermis of anther, which helps in the dehiscence of anther. Next one: pollen kit, a sticky covering found on the surface of the pollen that helps to attract insect. Next one: sporopollen, pollen wall material derived from carotenoids and is resistant to physical and biological decomposition. Next one: tapetum, nutritive tissue for the developing sporogenous tissue. So in this session, they are discussing about sexual reproduction in plants. That is male reproductive organs of the flower and andrecium in part 3 class. Next part 4 class, they are discussing about female reproductive part, gynecium. Thank you.